Hello everyone, I am Dr. Neena Vadera from Shivaji College teaching biochemistry. Today we are going to learn about amino acids. We will learn what are amino acids, what are the major classifications, and how to remember the structure of all these amino acids and classification. Now, how do we start? Why amino acids are important? They are part of proteins. Now, proteins are the most abundant biological molecules occurring in all the cells and all parts of the cells. Proteins occur in major variety and thousands of different kind may be available in a single cell. They are constructed from the same set of 20 standard amino acids which are covalently linked by peptide bonds. All proteins differ in their chain length, sequences, that is constituent, composition and sequences. Therefore, the structure of proteins is very important based on primary structure, secondary, tertiary and quaternary structure. Therefore, based on this they differ in their functions. Now, let us start with 20 standard amino acids. The basic structure of all the amino acids is the way we are going to learn. Why are they called as amino acids? Due to the presence of amino group and carboxyl group. Now, because amino and carboxyl group both are present, therefore, they are called as amino acid. Amino refers to amino group and acids refers to carboxyl group. Normally, we write NH3 plus and COO minus, that is ionized form in the. So, let us see how do we write. The simple basic thing to remember is we write C in the center, this COO minus group, amino group and H. This is basic. How all amino acids have this type of structure, and then here is your R group. Now, what is this R? R is side chain. This side chain differs from one amino acid to other amino acids and forms different types of amino acids. Earlier, the short form of amino acid was 3-letter words like glycine, we write G, leucine, we write L, and so on. But this way 20 amino acids will have all three letters, so it becomes a too lengthy for compiling the data. Later, 1-letter code was devised by Margaret Oakley Dehot. Who was the founder in the field of bioinformatics. This was done to reduce the size of data files for the amino acid sequencing. We can easily memorize and understand its origin. How was it originated? Like A, G, L, P, T. These are the basic amino acids which are present in most of the proteins, common proteins. A is alanine, G is glycine, L is leucine, P is proline and T is threonine. Another types of amino acid R, F, Y, W. R is for the arginine, F is for the phenylalanine, Y is for the tyrosine, and W for the tryptophan. Similarly, we have D, N, E, Q. Now, D is for the aspartic acid, N is for the asparagine, E is for glutamic acid, and Q is for the glutamine. Now, leftover K is given to the K letter is being given to lysine because very few letters were left. Why was it given K? Because K is close to the L, which denotes lysine. Let us move ahead. Coming to the proper classification. Let us see based on which type of R group is present, we classify amino acid into two major types. One is non-polar and another is polar. This is based on non-polar group attached to the carbon at the position of R, and polar where polar group is being attached to the at the position of R. Now, let us see how we remember the names of these. The amino acids are glycine, alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, and tryptophan. Now, how to remember these amino acids so that if any time we want to know which amino acid is polar, is being classified under non-polar. Now, if I see there 1, 2, 3, 4, 5, 6, 7, 8 and 9, it is very difficult to remember 9 amino acids. So, we can make a secret code word for them, that is G A V L I, that is G A V L I. Now, we write for this M P P T. So, how do we remember? For our convenience, we just remember Gavli MPPT. Please remember, these are not single letter codes given to the amino acids. So, anytime we want to remember that which amino acid, how many amino acids are there? So, we can say there are 9. So, and we can remember this code and we can assign them. But there you should remember that there are 2 P's, and which belongs to proline and phenylalanine. T is for tryptophan. Please remember, single letter codes are altogether different. Now, coming to polar amino acids. Polar, the amino acids can have uncharged group, groups, or we can say uncharged amino acids, or we can say that these are charged amino acids. Charged amino acids, I can write. So, what are charged amino acids? With charged groups. I can write here also uncharged amino acids which are uncharged, polar and uncharged. So, uncharged amino acids. or we can say neutral also. Now coming to what are the different amino acids? They are serine, threonine, asparagine, glutamine, tyrosine and cysteine. So how do we remember? There are 6 amino acids. So I can write here these are 9 amino acids. These are 6 amino acids. Again it is difficult to remember. So how will I remember? S for serine, T for threonine, A for asparagine, G for glutamine, T for tyrosine and C for cysteine. So my secret code to remember is STAGTC. Now coming to charged amino acids, they can further be classified to negatively charged amino acids or positively charged amino acids. Negatively charged amino acids, there are only 2. These are aspartic acid and glutamic acid. That is AG. So how do I remember? A for aspartic acid, G for glutamic acid. Similarly, there are positively charged amino acids. So these are lysine, arginine and histidine. So how do I remember? LAH. So here there are 3, here there are 2. So how will I remember? 9 non-polar, 6 polar uncharged, 2 charged, negatively charged and 3 they are positively charged. This is positively charged. So these negatively charged amino acids are also called as acidic amino acids. On the other hand, positively charged amino acids are also called as basic amino acids. So these things should be on your tips because these are the basics of any to learn the structure of protein properties. So properties of the proteins are based on amino acid constituent amino acids which are present in the protein. If there are more number of non-polar, then the protein will be more towards insoluble. If it is towards the polar, then the chances of soluble protein will be there. Okay. So we have learnt how to remember the names of amino acid under various categories or classification. In the next class, we will learn how to remember the structures of these amino acids. Thank you.